Hello, welcome to my talk today. I'd really like to thank the organizers of this conference for bringing together scientists and students, in this strange time where we're social distancing and many of us are still working from home. I think it's really important to keep each other engaged and to learn about all the great science happening within Canada. And so it gives me pleasure to talk today about the importance of using whole genome sequencing to track bacterial pathogens in poultry. We talk about the poultry industry. Chickens are obviously the most common bird on the planet, thought to represent 70% of bird biomass. And there are an estimated 29 billion broilers worldwide, which is truly an astounding number. I couldn't believe it when I read that. In the US, their industries consists of about 9 billion broilers, and chicken is the #1 meat protein consumed by Americans. And in Canada, the number of broilers is obviously less than 9 billion, but it's still significant. So when we think of chickens and eggs, we often think of diseases of humans, such as Salmonella and Campylobacter, but these don't really cause mortality in the birds themselves. The diseases of chickens, which do cause mortality and affect producers' bottom line, the E. coli, Enterococcus, numerous viruses. So in the past, bacterial strains would cause infections, would be characterized typically by things present on their cell surface. And these would be the H, K, and O antigens. So this is something called serologies. When you talk about a serotype, that's what we're talking about. When you think of O157H7, it relates to these antigens. More recently, then it's moved towards genetics with PCR. It can be non-specific, like a rapid type of test, looking for relatedness between strains, or specific, looking at toxin genes. So with E. coli, we know that the presence of Shiga toxin is strongly correlated with human disease. And pulse field gel electrophoresis was also used. PulseNet Canada was a group. So here's just a picture from a highly cited paper in 2006. And you can see that the patterns generated by pulse field gel electrophoresis, you can tell that strains are related to each other. What you can't tell from this is the individual nucleotide differences in some cases, or whether, basically just how related strains are to each other. And that's where whole genome sequencing comes in. We have the ability now to generate the complete complement of genes and understand it, the gene order and the specific DNA sequences, which allows comparison between members of the same species. So you're looking for tracks of genes that are in common or identical strains, which is a few small changes. And we use this for tracking the human hospital-acquired infections, and even with COVID, right? When we think of all these variants, it's all based on the genome sequence. And here's an example of how this worked with E. coli in a human situation. When you think of the German E. coli outbreak in 2011, was caused by a pretty rare strain of E. coli called O104H4, which is actually something called an enteroaggregate of E. coli. And what was unique about it was this causing hemolytic uremic syndrome, which is typically only associated with like enterohemorrhagic E. coli and the Shiga toxin. And so within a month of this outbreak, they had gained the whole genome sequence of this E. coli strain and realized that it's sort of this weird hybrid. Strains, Shiga toxin would not normally be found in this type of E. coli. So within a month, they're able to determine this, which helped future treatment. And it also was used to definitively identify the source of this outbreak as contaminated fenugreek seeds. So for our project, we're looking at colibacillosis. So colibacillosis is the #1 bacterial disease of poultry, causes morbidity, a mortality up to 20% in some flocks, and a drop in meat quality. It's caused by E. coli. Now, E. coli is interesting because we also have E. coli in our gut. It's a well-studied organism. It's a pathogen of humans and animals. And the strains that cause colibacillosis have been characterized as avian pathogenic E. coli. And they're diverse strains, but with specific sets of virulence factors. And the dogma here is that apex strains emerge from the commensal gut bacteria, so within the chicken itself, and it starts as a respiratory infection, often a secondary infection, just showing here in the lungs. So our questions related to colibacillosis are, is this a farm management issue? Is it really just due to bird health? So for example, if the birds become immunocompromised, perhaps through other infections, then the E. coli that's resident in them perhaps are the strains that come and cause disease. And related to that, are there outbreak strains, or is it just specific to each chicken? And so what we wanted to learn through a project is the relatedness of E. coli strains between Saskatchewan farms. Could you have an outbreak strain on two different farms? We want to learn about colibacillosis. Is there a difference between disease-causing E. coli strains and strains that come from healthy birds or from the gut of birds? And what are the levels of antimicrobial resistance within the E. coli strains? And so what was done in the past was producers would bring their dead birds in and interact with members of the poultry extension team at the U of S. They would do necropsy on these birds, send organ samples to Prairie Diagnostic Services, which is also basically like right next door. And they would be the ones to determine if there's an E. coli infection. If it was, that got reported back to producers that it was an E. coli infection. And so when I started to inquire about colibacillosis, I asked, where are these isolates? Have they been stored anywhere? Is there years of these isolates stored in a -80 freezer? And amazingly, there wasn't. And I knew there's a big opportunity for microbiology here to help investigate and learn more about the disease. And so what we are doing now is same process. So that the producer still brings in the dead birds to the poultry extension team. It still gets diagnosed. Now, when after E. coli diagnosis, a positive diagnosis, we've asked poultry extension to save us half of the internal organs. And at the same time, then go back to these farmers and say, okay, you've had an E. coli positive infection on your farm. Can you supply us with healthy birds out of that same flock? And through this, we want to compare disease-causing E. coli to healthy E. coli, so cecal E. coli. And so the systemic E. coli from the dead birds, isolate out of the internal organs. We'll do this now in our lab, as well as the healthy birds. We're sampling the cecal contents to get these kind of disease-causing versus healthy. And what we are doing is something called nanopore DNA sequencing. So this tiny little device here held in my student Madeline's hand is actually quite easy to run and cheap to set up. And it generates really long reads of DNA. And this makes it easier to assemble the complete complement of genes in the gene order. And Nanopore as a company is working hard to automate sample preparation. So I think their vision is to have these little devices be present right on a farm, so a farmer could perhaps do sampling themselves and put it right in here, where this would be linked to a researcher. And so the technology itself, I'm not going to bore you. It is actually a nanopore, a pore that the DNA gets sucked through to get DNA sequence. There's 3 YouTube links here, which can tell you how the technology works if you're interested. And so our results so far. So the first thing is looking at the whole genome profile. So each one of these circles represents the chromosome of an E. coli strain, the complete complement of genes. And you can see that the systemic E. coli from an outbreak look to each other. Remember, there's like up to 3 birds. And then the cecal E. coli is here in yellow. And so we've analyzed 18 different outbreaks so far, and we've collected over 500 different strains. And in each outbreak, the systemic and cecal isolates look different from each other. And what this tells us then is that the strains that are causing the disease are not coming from the intestines of healthy birds. So we're still compiling the final genomes, but they, in each case, have distinct regions of difference. So in these 3 here, if you're looking at spaces here, which represent tens to hundreds of genes that are missing compared to systemic E. coli. So the second thing I want to share with you, and what we thought, could you have an outbreak strain on 2 different farms? And so what we had is in 2 different outbreaks, we identified identical 078 H4 strains. These outbreaks are 4 months apart. It could suggest a common point of contamination, such as feed. It also opens up the possibility that these strains could be transferred between farms. And I should point out that the poultry extension members know which farms these came from, and we don't, and we want to keep it that way, at least until our analysis is done. The third thing is really, sometimes you enter into these projects and we're focused on the chromosome. There's more of the genome than meets the eye, because these E. coli strains also have mobile DNA elements called plasmids. And in many cases, they're a source of antibiotic resistance. So the first 100 isolates we analyzed, the systemic isolates or disease-causing isolates had a higher number of plasmids compared to these cecal isolates that come from healthy birds. And when you apply this to antibiotic resistance, and you can see the systemic isolates, 71%, were resistant to more than one class of antibiotics, whereas with the cecal or from the healthy birds, it's 30%. And a lot of the genes responsible for these differences lie on these plasmids. So our initial thoughts and observations, we expanded the project to start collecting environmental E. coli strains. So you can see here some of the location of some of these samples now that we're taking from barns that poultry extension members are getting for us. Questions: could pathogenic E. coli be hiding out in barns and then the birds pick up these strains? And this is actually the source of the disease-causing E. coli. And could there be matches between environmental and disease-causing E. coli, being that these are coming from the same barns? So this seems a bit like a fishing expedition, but we know that whole genome sequencing should be able to answer these questions. Like if two isolates are related, we will know for sure. So to summarize, what I've shown today and talked about is about colibacillosis. Now it's just one example, one pathogen, showing how whole genome sequencing can be used for tracking. We want to understand, and we will understand more about the disease. Could we identify potential source of E. coli, levels of resistance, which could ultimately lead to better treatments? And it's incredibly powerful technology. So for poultry in general and other poultry pathogens, we could have Canada-wide tracking. We know in many provinces there's common sources of eggs and feed, etc., other points of entry into a farm. And the other part is, could we get an overall feel for the antimicrobial resistance that exists within the E. coli strains on farms? And this can lead to the construction of databases. So the last thing is the acknowledgements, all the people that make this possible. So a lot of the work is done by my master's student, Madeline McCarthy. She has a poster here. Please go and look at it. Rosemary is a new student. Her job or her project is to look at the environmental E. coli's. And then Lindsay and Siva were part of this process. My collaborators, Dr. Joanne Dillon, who's also at BETO and the Biochem department, Dr. Rima Singh's a bioinformatician who's helping Madeline construct the genomes. And then Joseph Rubin is helping us with antimicrobial resistance testing. Michelle helped us there. Any of this is not possible without the poultry extension team. So Dr. Tyra Dixon, who's a poultry veterinarian for the university, Tennille and Nikki. And the funding here, and here's our COVID lab page. And thank you very much for listening. I'm happy to answer any questions.
Thank you, Dr. White, for this very interesting presentation. I have a question regarding the cost of such analysis and how widely available it can be made to the various diagnostic labs.
Hi, okay, can you hear me?
Yes, very well, thank you.
Okay, yeah, the cost, the nanopore MinION, the little device there is actually only $1,000 to get started. Each flow cell is where you load your samples. From one flow cell, we've been getting 24 E. coli genomes, and it's about $1,000. So it works out. It's pretty cheap, and the costs are continually coming down. So it's actually not a barrier. It's not like you have to buy an aluminum machine that's $500,000 or something like that.
Yes, yeah. This is quite amazing, and definitely opens the door to easy investigation of certain cases. You've given a great example with E. coli being probably one of the most expensive diseases in the poultry industry, but there will be also other pathogens too, which will follow. I have a question from the attendants regarding, well, first a comment, nice presentation and efforts. Nanopore sequencing is still prone to errors in sequencing. How do you overcome that to correlate the strains?
Right, and great question. Yes, Nanopore does have errors, but what it's amazing at is getting long enough pieces of DNA that you can create the chromosome. So the sequence isn't perfect there, but that's when we do Illumina sequencing, so short read sequencing. But because we don't need to de novo complete the genome, we just need to fill in the gaps from nanopore, we can really scale it up. So in there we do 96 strains at a time, and that enables us to get perfect sequence, basically.
Okay.
So it's a combination.
This would work then for ideally for pathogens with known sequence, but for those who have an incomplete sequence, this probably wouldn't be the ideal tool.
No, it's that's the beauty of nanopore. The long read, C. coli has a lot of repetitive regions in the genome, so we are really afraid how we're going to get a complete circle, because if you do short reads, it's very hard for a computer to stitch them together properly in a proper order. And that's where nanopore, the combination of the two, actually makes it so you don't need to know the genome of this organism. Okay. You can use nanopore to make the scaffold, and then Illumina to fill it in.
Okay, well, as mentioned, the attendee who asked the question, this is a very smart approach.